Okay, hi everybody. So welcome to our webinar about the MSc Regenerative Medicine. My name is Dr. Kristen Brown, and I'm one of the course directors, and my colleague, Dr. John Connolly, is also here.
Hello.
Hi, John. We didn't rehearse that, did we? So what we're going to do today is give you a little overview of the program and discuss a few research highlights. We did give a webinar in May, and I'm not sure, actually, if you mind putting in the chat, did any of you attend the May webinar, just out of curiosity, or is this your first time? You could just put in the chat if you were here before. It'll help us a little bit. Okay, so Laura was there and Jishan was, saw the recording, which is good. So what I think I'll do today is I'll do a brief overview of the program just to kind of direct questions from you guys. So that will be a little bit of a repeat of what we did in May, but I'll try to go over it a bit more quickly so that we can, I think the most important thing today as we're getting closer to the beginning of the academic year is to give you the chance to ask us questions. Okay, so the format of the webinar is I'm briefly going to do an overview of the program, and then Dr. Connolly is going to go through some of the research highlights from our staff. So we always like to start with a slide of what is regenerative medicine, because it is something, it's a word that term that's thrown around a lot, but actually has quite a lot of different definitions. And this is one definition that we really like. So it's the use of cellular or biological components to regenerate damaged tissues or organs. And it brings together a number of fields of study, and that includes basic biology research, especially stem cell biology, but also biomaterials and tissue engineering, and finally, clinical medicine. So in particular, in developing preclinical and translational applications for patients. So the design of our program, the MSc Regenerative Medicine, we've been running the program since 2014. And actually, we're just in the midst of doing a program review to make sure that everything is still current and up to date for you guys when you come in. But we have a full-time one-year program. It is interdisciplinary, so it is a collaboration between the School of Medicine and Dentistry and the School of Engineering and Materials Science. The program design is that we have 8 taught modules, and each of those modules are 15 credits, so that makes up 120 total credits. And then we close the year with a dissertation research project, so that is worth 60 credits. In terms of the structure of the program, so we start the course in September, and we start with the students taking 4 modules, and these base modules are molecular and cellular basis of regeneration, stem cell and developmental biology, research skills and methodology, and advanced tissue engineering and regenerative medicine. And we have all of our students take these modules because they think they form a very good foundation for the course. When Dr. Connolly speaks to you, he's going to highlight some of the research of some of our leads for these modules as well. Then in the second term, which starts in January, well, at the beginning of January, students have exams from semester 1, and then they start term 2. And students have the option of choosing 4 out of 5 modules. And the choices are ethics and regulatory affairs, which is offered by our colleagues in engineering, biomaterials and regenerative medicine, tissue-specific stem cells, induced pluripotent stem cells and genome engineering, and neurodegenerative disease. So students choose 4 of these 5 modules, and we can help with the choice. Usually students get an idea during semester 1 of the things that interest them, but the students will have advisors and can obviously talk through their interests with their advisor and to help them make the choices that are best for their training. So we close term 2 in around April, and then students have some exams again. And then we start the research projects at the beginning of May. And the research projects are an exciting time of the program. So students join our labs full time. They really become members of the team, go to the lab every day, join into lab meetings, present their work to the group, and get the same kind of training that like PhD students get in the lab, this full-time use of the equipment and supervision. So it's a 12-week research project. How we do this is we call for projects once we know the number of students, and we allocate or distribute them to all of our students in October, so a little description of each project. And then the students have the chance to meet with supervisors during the first term, and they can submit a list of their preferred projects. Usually we have students rank their top 5 projects out of the 20 or 30 that we offer, depending on the number that we have in the course. And then we just simply go through a process where we try to assign as many projects to students where it was one of their top choices as we can. So we don't base it on grades or anything. We just try to put as many students in one of the projects that they wanted as we can. And usually people get one of their top 2 or 3 choices out of those 20 or 30. And then the work in the labs begins in early May and continues through the summer until August, when students complete a final dissertation and oral presentation of their project. So these are just a few examples of research projects that our students have completed in recent years. And on the right-hand side are some really nice data that our students have generated using flow cytometry and confocal microscopy. So you can see the quality of the data that they produce and the access to really good equipment to generate that data. So a few examples are genome-edited stem cell models for severe obesity, 3D printing of vascularized human skin equivalents, cerebral organoids from living patients as a model to study brain circuitry disruption in aging and Alzheimer's disease. That one sounds really interesting. And ECM production in next generation bioreactors for tissue engineering. And you can see here that we've got a good mix of projects that are more biology based and also a bit more engineering based. And that reflects the fact that we place students not only within the Faculty of Medicine and Dentistry, but also within the School of Engineering and Materials Science. So what are our plans for next year for teaching? Our teaching and learning will be delivered via mixed mode education. So what does that mean? So we're planning our activities to be on campus. So lectures, discussions, student presentations. This is a small program, so it's very interactive. And we have the students do a lot of group work and work with each other and present journal club papers. So they develop a lot of presentation skills. And then we supplement those on-campus activities with the asynchronous online activities. So maybe additional reading or some videos to supplement the learning or some quizzes that you can complete after a lecture to kind of help to reinforce the information. So when we say mixed mode education, that means that students do have the ability to log on to taught sessions remotely if necessary. But we always encourage students to come to the on-campus sessions if they can, because the interaction with the lecturers and with each other is really an important part of the course. But if you're feeling unwell or there's a reason you can't come to campus, we do have this option of being able to log on and keep up with things, or in fact, to watch a recording afterwards, because if you're sick, you should just rest and watch the recording. Our exams will be delivered online. So we have open book exams that will be completed over somewhere between 4 and 24 hours. We try to write quite challenging exam questions, so they obviously can't just Google the answers, but have the chance to really apply their knowledge in a real world kind of way. And then just a reminder that we do expect that full-time laboratory projects, thesis projects will be starting in May of 2023. In fact, it was only during 2020 that we had to suspend our lab projects because of COVID. We've been back in the labs since autumn of 2020. So very briefly, what do our students do after the master's? First of all, we're very proud of the number of students who've gone into PhD studies, both in the UK and internationally. We've had about 30% of our graduates have progressed to PhDs. In fact, we just heard of another one today who's been offered a place. And I just threw together a slide quickly to show you some of the places in the world, in the UK and in the world, where our students are now undertaking PhDs. So we've got a couple in Japan now, and South Korea. We've had a few go to Spain, Germany, Greece. I can't remember where these all are. The University of Illinois in Wisconsin, in the United States. And then in terms of in the UK, or the British Isles, I should say, University College Dublin, Aberdeen, Cardiff, Lancaster, Manchester, Cambridge, and a number of the London universities, Queen Mary, King's, UCL. So we've had really good success of our students progressing to PhDs following completion of the master's. And we think that's a reflection of the training that they get with us. Some of the other things that students do after their master's are go on to become research assistants, progress to roles in industry. We have a number of students who want to go to medical school, and we've had, I think, 8 students now who've gone to either veterinary or medical school after completing the master's. We've had students who've gone into science publishing, teaching, and a number of other kind of science-associated careers. And to support our students in these career choices, we have a couple of things. We have a careers day where we bring in both alumni and our network of collaborators to speak to students about opportunities. But we also have a LinkedIn alumni network. And Queen Mary offers really good support from Careers and Enterprise. So they support students not only just when they're here, but also for 3 years after graduation, and helping them to prepare their CVs, prepare for interviews, and find jobs. Here's a picture of some of our lovely graduates from a few years ago, a number of whom have actually now gone on to complete PhDs and have moved to the next step of their career. So I think what we'll do is I'll pass over to Dr. Connolly, and then we'll take questions at the end of the session. If you want to ask something and you're worried you'll forget, just put it in the chat and we'll catch up with you a little bit later. Just a closing shot. This is the Blizzard Institute where the program is based, and this is what it looks like at night. You can see it's a very beautiful and progressive institution, and the equipment that we have is excellent that our students have access to as well. So thank you for your time. I'll pass over to Dr. Connolly, and then we'll get to the Q&A, which I think is an important bit.
Thank you. I will just go ahead and share my screen now. Okay. So I think, Kristen, does that look all right?
That's perfect.
Okay, great. So what I thought I would do this time around was give you a little bit of an introduction to who we are, who you'll be taught from on the program. So last time I covered some of the big research findings or broad areas of research going on at Queen Mary. But today I wanted to focus a little bit more on the scientists who are going to be actually delivering the course. And I think that's important because, we're not just lecturers and educators, but we're also active research scientists working within the field. And so the three that I'd like to highlight today is work that we're currently doing on image-based profiling of senescent cells, which are hallmark of aging, the epigenetic regulation of different types of stem cells, and then actually a little bit of work from my own lab where we're developing 3D printed models of normal and wounded skin. So first off is work being done by Dr. Cleo Bishop, who leads the cellular and molecular basis of regeneration in semester 1. And the focus of Dr. Bishop's research is to try to understand how cells and tissues age, and in particular, the process of senescence. And some of the recent work from Dr. Bishop's lab has been developing this image-based method for profiling and defining the properties of a senescent cell. And so for this, they've been using different types of automated microscopy and imaging, as well as computational studies to analyze these different morphological changes in the size and the shape of the cell and how that changes when they become senescent. And so this is a recent publication from their group defining these profiles based on imaging, which hopefully will be an important tool for further research into mechanisms of cellular aging. And then another bit of recent research, this time it's from Dr. Miguel Branco's group. And so Dr. Branco leads the stem cell and developmental biology along with Dr. Brown in semester 1. And so Dr. Branco's group is primarily interested in what's referred to as transposable elements. And so these are regions of DNA within our genome that move around a lot, but don't encode for genes themselves. And so for a long time, there's been this sort of mystery about why are they there? Do they do anything, or are they just junk? And it turns out they are really important. And so in this recent study from Dr. Branco's lab, they're looking at how certain transposable elements referred to here as TE regulate the expression of genes that are important for maintaining different types of stem cells in their undifferentiated state or directing lineage-specific differentiation. And so Dr. Branco will be providing more insight into these developmental processes and fundamental stem cell biology. And so he's really an expert in the field and can provide this state-of-the-art insight. And then lastly, there's some work being done by my lab. So I am a bioengineer by training, and my group is interested in skin repair and regeneration. And some of the work that we're currently doing is to develop new hydrogels and biomaterials for promoting skin regeneration. And so here we've developed a panel of extracellular matrix derived hydrogels that we can use to mimic the dermis of the skin. And some of our recent studies have looked at the composition and structural properties of these and how they can be used in vitro to recreate a very functioning, a high level of differentiation and function within our skin models. And that's what's shown here on the left. In addition to that, we've also been doing some work trying to develop materials that can be 3D printed so that we can create 3D printed models of either intact skin or, as shown on the right here, a skin model that has a defect in it so that we can then study the process of wound healing and tissue repair. And so we'll be studying some of these same principles within the biomaterials module that I lead in semester 2. So that was just a very brief kind of introduction and snapshot of some of the work that the lecturers on the course are involved in. But really, what I'd like to highlight with this is that the course is delivered by not only experienced educators, but also research active scientists. And therefore we have access, the topics that are going to be covered across all the different disciplines are at the forefront of regenerative medicine research. And as a result, the students will have access or the opportunity for state-of-the-art training in these fields. So as Dr. Brown mentioned at the beginning, we wanted to keep the intro and highlights a little bit brief so there was more time for questions. So what we can do now is to switch back to, we'll stop sharing. So we'll now open it up to questions from the group. So feel free to either raise your hand and we can speak to you directly, or put your questions in the chat.
All right, so Jishan, you're a MBBS student at Bart's at the moment, if I remember right. Is that right?
Yeah, that's right. Yeah.
Okay, fantastic. It's nice to have you here, first of all. So the exams you'll find are quite different than medical school. So they're online, but what is the format? Is it essays, multiple choice questions, short answer questions, or a mix? So they aren't multiple choice questions. So at master's level, we don't tend to use multiple choice questions, but we do use essays, some short answer questions. So things like data analysis. So students are given data and, kind of have to be able to interpret that data and ask short questions around that. So those are the two most common forms of online assessment that we have. What's the second part? Sorry, it's lit up on my screen.
About accommodation. So I can respond to this if you'd like. So in terms of contact hours, I think each module has about 2 or 3 contact hours on campus per week, and that will be most days of the week. I think for this upcoming semester, there's scheduled contact time every day of the week. Some days are much busier, and you'd be on campus almost the whole day. Other days it might be a half a day. So, depending on, how you would like, the commute is for you, you might want to be closer or further away from campus. And then, of course, in the 3rd semester, when you're doing the research project, you'll likely be on campus most days and for a full day.
All right, thank you. That's very helpful.
Okay, great. I can do the next one as well, because that's about the bioengineering. So Monty asks, someone who hasn't studied engineering before, but is interested in the bioengineering part of the course, are there ways I can prepare from now until the course starts so I'm more prepared? So I'll answer this first bit. So this is a common scenario for students. So there are engineering components to the course, and many students are not engineers by training. So we do a couple things to help that transition. We do run a couple additional sessions in the first semester. I actually lead those, introducing students to essential concepts in engineering and mechanics that they'll need for the tissue engineering module, but later on. So I wouldn't say there's anything that you need to do now, but there will be some additional revision or work that you can do once the semester starts, but we can help guide you to those resources. One other thing I would say along those lines is that because this is an interdisciplinary course and we have students from all different backgrounds, no one on the program is going to be an expert in everything. So for everybody, there will be some point in the program where they're encountering something that might be new, unfamiliar, or particularly challenging for them. And that's natural. And but that's, I think, part of what makes the course exciting, is this opportunity to learn new and different things. And so, we acknowledge that, and we're, happy to support you. And also in the past, the student peers have supported each other. So if someone has a clinical background or an engineering background, they're happy to help each other out, and they can learn from each other, which I think is an important benefit.
Yeah, I think the other thing I would add to that, those are all very good points, is that Dr. Connolly and I review all of the applications, and everybody who's applied this year, we've had students with similar backgrounds in the past. So we've had dentists, we've had veterinarians, we've had medical trainees, we've had clinicians who'd finished their degree years earlier. We have the most common background is kind of biomedical sciences or biotechnology or those types of degrees. We've had bioengineers. So we have a lot of experience in supporting students. And I think it's a really important point that everybody will have an area that's a little bit more challenging for them. And actually, I think that is what makes the course interesting. I love how students, especially in group projects, they learn from each other and work together. And certainly I learn a lot from the students too. I know nothing but engineering, and I always learn a lot from the clinicians, and that's really exciting for us as teachers as well. Yeah.
Do you want me to grab the next one? I think we're on, will there be any opportunities to progress onto a paid PhD after the course? So there are opportunities within Queen Mary or other universities, but they're not directly connected to the course. So the course doesn't feed into a specific PhD program, but there are PhD programs at Queen Mary and other studentships, and the students who have gone on to PhDs have applied through those. So there are opportunities out there, but they're independent from the course.
Okay, so the next question is, as we're starting in labs, how long do we have to gather data, interpret our data? So Maria, that's a good question. So the lab-based projects are 12 weeks in duration, but something we try really hard in the course to protect is 12 weeks of actually being in the lab and working. And then we give the students an additional amount of time, I think it was 3 weeks this year, to write up. Now, the students should be writing up as they go, but we think it's really important that 12 weeks in the lab is protected so that students can generate as much data as they can and interpret data. We also, students get a lot of support from their supervisors in terms of interpreting the data, but we also do some webinars about, so you just did one this week, didn't you, John, about how to generate figures to show your data effectively. We do seminars on how to write up your dissertation. So there's support, for the students throughout that process. And I think it's also worth mentioning that the biggest points of emphasis, I think, on the course are the ability to analyze data and present data, and to present well, both in written form and orally. So we emphasize that throughout the year. So how to analyze data, you'll be starting really from the first semester, and we'll be starting with certain techniques and emphasizing that. And so hopefully by the time you get to the project, that's something that comes a bit more naturally to you. I hope that was clear, Maria. Feel free to follow up with a question if it wasn't. John, do you want to get this one?
So I think that, would it be difficult to, would the time frame allow for projects that would enable us to get directly involved in cell biology, culturing and screening techniques? Can we get involved in the clinical aspect of things? So this is all going to depend on the specific project that you choose. So Some projects might involve cell culture and screening and much more in vitro studies. I think the majority of them are kind of in vitro based and involve cell culture. There are some projects that involve more in vivo work, either with animal models or other organisms. And then I think from time to time, there are projects that have a more clinical slant, whether it's working with clinical samples or developing diagnostics, or potential, or more on the engineering side of things. I think it's unlikely that you'd probably be involved in something like a clinical trial, because those are usually very organized and controlled, and not so amenable to a master's project. But there might be clinical research going on within the group where you would be working. But it all depends on kind of what projects are available that year and which project you would choose. And then maybe just to follow up on, would it be difficult to fit in with a dentistry background? Nope, I don't think at all. We've had a number of different, we had a number of dentists on the program, and they've all done very well. I think the dentists tend to do well coming from a clinical background, but also the materials. Many of the dentists are familiar with lots of different dental materials that are relevant to tissue engineering. So that aspect they usually do quite well. Kristen, do you want to take the next one?
Yeah, so the next question is, what are the clinical prospects of this degree in our future career? Mary, I'm not entirely sure what I understand what you mean, so feel free to either jump on mute or just type a follow-up question. So we have had a number of clinicians on the program, so both dentists and one veterinarian and medical doctors. And I said it's important to emphasize that we aren't doing clinical training on this program, but the people take away the training they have in this program to kind of take the next step of their career. So some have returned to clinical practice. So for example, we had a dentist a few years ago from overseas who's now doing a PhD in the UK, a dentistry, regenerative medicine associated PhD. So they're, I think it's a step. The knowledge of the program can be used in a number of different ways. But one thing we don't do is we're not doing fillers or anything like that practically. No, it's not a clinical program.
I think maybe just to elaborate on that, there have been a number of students who have come from a science background, and following this course, they have gone on to apply and be accepted into medical school. So that is an additional career route for the graduates. But as Dr. Brown said, we're not providing clinical training here, it's scientific training.
Yeah, and I think just one more example is kind of coming to me as so we have also had intercalating students in the past who had an interest in kind of going down the academic fellowship route, and we've had some success of done this master's going on and getting accepted to those very competitive careers, and probably will go on to do an MD or PhD after that. So it kind of depends on where you're starting from, your background, in terms of different ways to use the degree. But I think the skills that we teach are quite transferable to a number of different pathways. Does the course contain any clinical application of skin diseases as part of the dissertation, for example? So we have real strengths around skin biology. Both John and I are actually in the Center for Cutaneous and Cell Biology, cutaneous research and cell biology. So we have a lot of people working in both basic skin biology and coming on to some diseases within the skin. So there'd be the opportunity to do work in those types of projects. Again, probably more at the kind of in vitro, foundational level of knowledge, as opposed to kind of translating into the patients. But something else I should emphasize, I think, is that there are a number of opportunities on the program where you can dig deeper into things that interest you. So there's opportunities to do literature reviews. On John's module, there's an opportunity to write a grant where you use biomaterials to address a problem. So you can pick the problem that you want to address. So if you're interested in, say, skin biology, there's a chance to go deeper in that topic. And I think it's really important at master's level that you can kind of, within reason, tailor the course to fit your interests. John, do you want to add anything to that?
Nope, I think that sounds good. I can pick up the next one. So Maria asks, would there be opportunities to getting published or further research after the master's? So yeah, this is a really good question. I think for the most part, because of the duration of the project, the most likely scenario is that the student would do a bit of work that would become part of a larger project or publication. And so they might be a co-author and might contribute a figure or part of a figure of data. But it's unlikely that they would have a first author publication, or the master's research on its own would be published. We did have one example of that. So one student had a really good project and was able to get that published in its entirety as a first author. But in most cases, and we've had a number of other examples of this, the research that the students do is one part of a larger study, and so they would be a co-author on those papers. And then in terms of further research after the master's, this all depends on projects and funding. So oftentimes professors or PIs will have little bits of funding or need help with a project. And if the student working with them has already been trained and experienced, they might have an opportunity to continue doing that research. So for example, I had a small amount of grant funding a number of years ago, and after a project finished up, one of the master's students continued on and worked on that for 9 months. But it all depends on kind of funding and timing, and when it gets to the end of the project, seeing what the opportunities are.
I think Mona's question, John. I think it's better for you.
Sure. So would it be hard to understand some specific technical bioengineering topics for me as a dermatologist? I've never studied about such topics. So it will be different and it will be challenging, but it is completely doable. I think all of the bioengineering topics are approachable for someone who has not had a bioengineering background. We provide training and instruction in the kind of fundamentals that you will need to understand those topics, and it is perfectly within the scope of the master's program to learn about those if you haven't had any background.
Yeah, I think just to pick that up a bit further, Mona, we find often our clinicians at the beginning of the course can feel a little bit overwhelmed because they're learning new techniques and things that they feel like the biosciences graduates are more familiar with, like Western blots or PCR. And we have a program to really help people to catch up. But we also find by the end of the program, our clinicians tend to do very well. So it's kind of having faith in us that we've done this before, and we know how to build up that knowledge over time, so that students end up doing some really excellent dissertations at the end. And the whole idea is coming in to learn new skills. So it is that challenging yourself and trusting the program that we've done this before. I'm curious. So one of the things I'm proudest of with both the university, Queen Mary, but also our program, even though we're not a large program, we tend to be quite diverse. Would you mind, if you don't mind disclosing, would you mind just typing where you're from and what your previous background is, or your degree or your training? And we'll pick up some questions after that.
So maybe while the other ones are coming in, we can answer this next question. Are there opportunities to work as a lab assistant? Yeah, I mean, similar to the previous question about further research opportunities, there are often research assistant jobs coming up kind of at the end of the program, and students are free to apply for those. And I think based on the skills and knowledge that you've acquired, can be quite competitive for that. So I mean, there've been a number of students who have gone on to work in the labs where they did their project, but then there've also been a number of students who have gone to other universities or research institutes to work as a research assistant.
Okay, so we got a nice mix, quite a few clinicians actually. Dermatology, GP, intercalated medical student at King's, intercalated medical student at Bart's. Biomedical Sciences, Monty, at Brunel. We've had, I think there's someone from Brunel in the course this year, in fact. So it's a really nice mix, and that's what we tend to see each year. And I think that works really well for the program, having this mix of backgrounds. It's something certainly I've enjoyed. And coming back to the question that you asked about engineering, or some of you asked about studying other fields, I think it was something we were worried about when we set up the program too, wasn't it, John? But we found that, you know, if students are given the right support and the right background, and we think we do that well. Our students from all these different backgrounds have done very well. Laura, I see your hands up.
Hi. Well, my name is Laura. I'm from Colombia. What my question about like being a lab assistant was like during the course, not after finishing, but if we can work during the course.
I mean, yes, it is technically possible. However, do bear in mind that the course is quite demanding, and it's kind of expected that it's a full-time commitment. That being said, we know that a number of students have part-time jobs outside of the course to make a little bit extra money. So, you know, depending on how you balance your time, it is technically possible, but I would also be careful and make sure that, you know, you are devoting enough time to the course, which is quite demanding.
Yeah, and I think just to add,
Okay, thank you.
so just to add to that, Laura, we're not in a position to find people jobs as lab technicians, so it would be something that a student would have to self-arrange with the lab. And kind of in accordance with UK law, all the jobs have to be advertised and posted. So it's not a question of, you know, where we can set our students. Well, that would be lovely, where we can set our students up with positions without going through the interview process. Are the exams at the end of the year or after each of the modules? So Maria, you're a medical student, Bart, so you're used to the end of year exams. So we don't examine that way. We examine, we have in-course assessments. So some of the elements are things like oral presentations or written papers. And we also have exams, and we examine at the end of each semester. So we have a set of exams in January, which covers semester 1 modules, and then a set of exams in April, May, which covers the semester 2 modules. And the theory behind that is to try to get all the exams out of the way before students really get embedded in their research projects. And I think that works quite well. But I know it's very different than the style that's used in medicine.
Great. Are there any, so we had a number of questions. Are there any kind of final questions before we start to wrap up? I'll give people just another second or two to get them in. That's been good. There's been lots more questions, I think, this time, probably as we're getting closer to the start of the year, the realities and are kicking in and people are thinking more practically about things. So that's good.
We're thinking about maybe doing one more webinar just before the start of the year. Is there anything that you guys as a group would like us to cover next time, or we can address that now if you want? When we start the course, that's a good question. So induction week starts September 19th this year. So during that week, there'll be a few induction activities, including one for the course and one for the institute. And Queen Mary has a number of things, for example, for international students. It probably will vary a little bit. So for those of you who are at Bart's already as students, you know, you probably won't need the broader ones. But we do recommend that everybody comes to the course induction, because we go through the nuts and bolts of the course and things like how you log onto our online platform and how you get registered for your modules. And it's actually just really nice to meet everyone in the course. So that will be the week of the 19th of September. Then we'll start the teaching the week after, the 26th of September. When will we be hearing back from admissions overseas, and a bit tricky for me on timing? Miriam, so we kind of roll through the applications as soon as we get them. I can't remember yours exactly, but what we can do maybe after this webinar is check on the status of your application. But I think we have, we've certainly offered all the ones that have come through to us.
Yeah,
So
I think we're up to date on the ones that have been sent to us by the admissions office.
if you don't mind sending us an email, Miriam, we can follow up on your application. John, do you mind doing that? Because I'm on leave from tomorrow.
Yeah, I can do that. I think there's one other question from Jishan. When would we expect a timetable? Would that be once the course starts or earlier? So the timetable would officially be available during that induction week. However, I think we are just now finalizing some of the room bookings. So if you did want a bit of a heads up about kind of what the times and days are of the week when you can expect to be on campus, we can send you that in advance, if that helps with sort of planning and commuting and that sort of thing. But the official kind of room locations, times, all those details you would get during induction week.
Okay, Miriam, thank you very much. Yeah, we're happy. Dr. Connolly will follow up on that for you. It's no problem.
Great. I think looks like the questions are kind of dying down. Is there any further information we need to give the students at this point? There's going to be a welcome pack coming out, which has a bit more detail on enrollment and getting started, and that's usually the first week of September.
Yeah, we'll send out so we'll send you out a few. So you can see David in the background. It's just put something in the chat. So David's our digital communications lead. And we will send out we'll start emailing you a little bit of information about the program. And then we will, the first week of September, we'll send you details around induction week. So you know exactly where and when you need to be for induction week and kind of what happens the first week of the course. So we'll, I think, maybe try to do one more webinar, but we'll try to communicate with you a little bit more in writing so you know what's happening. But please do, it's a small program. So please, if you have any concerns or questions, don't hesitate to email one of us and we're happy to answer your questions. Also, if any of you are still kind of not sure, about the program, we're very happy to organize an individual chat with you and kind of talk through your concerns or your questions. I know sometimes this type of webinar is quite intimidating, so that's absolutely no problem for us. In fact, we enjoy meeting students that way. Okay, should we wind up there? Thank you all for your time and great questions. Actually, they're really really helpful. It makes the interaction much more nice for us when there's a lot of questions to answer.
And thank you, everyone. It was nice hearing where you're all from, and yeah, hopefully we'll see you in the autumn, face to face.
Thank you, everyone. Bye-bye.
Bye.